first speaker is Dr. Kevin Matthew. Currently, he is a director of Cell and Molecular Biology at American Dental Association Science and Research Institute. He is also a founder of Stratica Biosciences. Goal is pioneering AI-driven precision medicine. This company is currently linking spatial biology to clinical outcomes for patient stratification to better guide clinical decision making for surgeons, pathologists, researchers alike. Also, he has many active collaborations with single cell and spatial genomics companies like BioImage, 10x, OmaPix, Acquire Biosciences, ACD, Biotechnic, and Dipcell. His vision bridging oral and systematic precision medicine has garnered support from industry foundations and federal grants. So today his topic will be the precision lens, high resolution spatial biology for healthcare solutions. Please, welcome, Dr. Kevin.
Thank you so much. Let me share my screen.
Yeah, you can share.
Can you see that?
Yeah.
Yes, it's visible.
Excellent. Thank you so much for that wonderful and warm invitation. Gopi, it's been great to connect with you virtually and in person over the last, I guess it's been over a year now, or right at a year. And so today what I'm excited to tell you about is really, I think, launching forward from some of the work that that Gopi just shared with all of you and some of the great progress that TheraCUES has made. And to tell you, I think, whereas they start to consider both single cell and spatial solutions, some areas where we see some real innovations and advancement for really taking some of these basic science technologies as I think they were first founded and starting to move them towards the clinic. With some of the vision that I think Gopi has shared, I think these tools are helping us make those innovations and some of those advancements. And so today we'll talk a little bit about the precision lens. And so what I see is an opportunity for linking, I think, some of this work to really systemic health and outcomes that are predictive and hopefully actionable in the future. As was mentioned, I do have some disclosures. I also serve as a scientific advisor in addition to being CEO of Stratica Biosciences. Some of the work that we'll share today is in collaboration with companies, especially some of those that are advancing the field of spatial biology. And so that will influence a bit of today's presentation. At the heart of our lab and where we work in Gaithersburg, Maryland, outside of Washington, D.C., is really a linking between both federal, academic, as well as industry partnerships. We've had federal funding and academic funding the same, both internal as well as some foundations. A lot of the work we're going to share today is across a number of networks and collaborative team science approaches. Some of the advancements that we've made have been in collaboration with some of the people that you see on the right side of the screen. And really, we couldn't have done this work without their immense contributions and sacrifice to advance some of this work collaboratively. We're excited about where the future goes, and we don't see single labs doing this work on their own. But in a lot of ways, the collaborative nature of many of these labs, not just in the U.S., but across the world, of helping to advance this for not just U.S. health, but hopefully for global health initiatives. And as was mentioned, here's some of the companies that have also partnered with us to advance this mission. And so one of the things that we'll talk about today is that we see a lot of opportunities in the spatial analysis software space. We started to use some of this for some of our work focused on infectious, autoimmune, and also cancer, and particularly in the upper airways. Our lab is primarily focused on upper airway and lower airway diseases. And really trying to understand some of the mechanisms that guide really structural cell fate. And so some of these publications that are both in preprint and abstract as well as published stays will influence some of the outcomes you'll see. And we'll highlight where those in process or publications are in the bottom right corner of the slides. And so as part of the initiatives that we'll also share today is that, in a lot of ways, this work was founded by the Human Cell Atlas's initiative to really comprehensively map all the cells of the human body. Currently within the Human Cell Atlas leadership, led by Aviv Regev and Sarah Teichman here on the screen, there are 18 bionetworks. And a bionetwork can be focused on either a body niche or a technology. There are organoid networks, but there are also networks of the lung, the skin, of course, the liver, and the upper airways, which we lead. This leadership has allowed us to take the data that we'll share today and to start to link that to really publishing at the end of next year the first draft of the Human Cell Atlas, which should hopefully be an equivalent publication to the Human Genome Project, essentially categorizing over 100 million cells across the human body. And so the members of our bio network, which I lead with in Sikira at Queen Mary in London, include members both in the US and, of course, abroad in many countries. I think currently we're on all continents except for Antarctica, and there are labs participating across, I think, 75 labs at least across the world. And so what this allowed us to do is to start to think about how it is that we could leverage toolkits, data, assays, and really get us to a place where we can start to apply and start to infer some of the patterns of what we call multiomics. So linking that clinical information and kind of those outcomes measures in patients to the way in which we profile many of the omics tools. That could be transcriptome, that could be proteome, even thinking about the way that we could profile the TCR, BCR, metabolome, and even epigenome, and linking that back to understanding not just what a disease is called as it's diagnosed, but some of the pathways, patterns, and genes that are affected kind of comprehensively in that individual. And so as we'll just focus it today for a moment in the upper airway, in this case looking all the way back to the tonsil as we open the mouth, one of the things that we notice, and this is being categorized across the brain primarily, also in the lung now, that there are many microniches. So we typically think of these tissue structures as being a monolith, being one site, breaking down in one person and kind of one oral cavity or one lung. What we're finding is that there's anterior-posterior diversity, there's site specificity for some diseases, and so in a lot of ways, the space in which a disease arises can also dictate its outcome. And so the lab that we're trying to build upon really is to understand both the cell types, the patterns and trajectories of their differentiation, their gene programs, the mechanisms that guide some of those breakdown events that might influence cells around them in a spatial context, really linking this all to kind of a tissue level biology. And so if we're going to eventually move forward with understanding kind of the micro niche, we also need to model it. And so one of the things that we're finding very quickly is that as we make these in-person human discoveries, that some of our cell cultures, organoid models, primary cell lines, animal models just don't have the cells in the arrangement to make the inferences we need. And so we're working a lot on trying to understand how we can better fit the right disease question and the cellular makeup to the models that we have, and if new models are required, we're trying to build those. The goal of that, hopefully, is to make it such that an inference that is made goes back to clinical trials in a really kind of a shorter timeline with a higher chance for success, because we know many clinical trials fail as they move towards final approval stage. And so the other thing I'll say is that the oral cavity oftentimes gets, quote unquote, overlooked as one of the sites of importance. What I'm showing you here is a bunch of manifestations of inflammation and tissue breakdown, but this is not an oral disease. This is happening, unfortunately, in someone that has chronic inflammatory bowel disease across a variety of sites and conditions here. What we know is that it's bidirectional, the linkage of these breakdown events, both in the oral cavity and systemically, but also systemically and influencing the oral cavity. And so the real key of the Human Cell Atlas is that we get to integrate our data sets with the Human Cell Atlas overall and start to understand kind of the linkage events of kind of how disease can seed in one site and move to other parts of the body. So some of our first work on a project that we're hoping to preprint here in the hopefully next couple of weeks, is that we've linked all the data sets and also some new data sets across these various micro niches to kind of understand at the earliest phase how it is that gene programs, cells, and what we call modules. That's a cell expressing certain, let's say, chemokines or other effector cytokines to kind of guide overall cell behavior and sensitization. And so as we link both the pathology of all these distinct micro niches, we started to move forward on single-cell RNA sequencing, in which we have integrated both published and new data. We started to integrate and move forward on a highly multiplex immunofluorescence atlas, in this case using a 44 antibody panel, and then linking some of our software I referenced before on understanding cell types and what we call neighborhoods, the kind of arrangement of cells and patterns in tissues. I will tell you that my early ideas were that the cell was oftentimes guided by what we call intrinsic behavior, but I'm learning more and more often now that the cells around are influencing oftentimes the cell's behavior extrinsically, more often than I thought. That's also something we started to profile using highly, let's say, highly plexed-ish or in situ hybridization. These are probe-based assays that detect RNA at this current stage. and then integrating all those analyses computationally to start to understand how cells talk to each other, and performing kind of a receptor ligand analysis at scale, considering the niches as well as disease states. And so I'll say that in some of that work, and I'll summarize a long project with a lot of inferences we've made, but we're finding that the oral mucosa in itself might serve as it's kind of a nascent site for innate immunity, and really harbors an activated innate immune population and adaptive immune population, as we're kind of recently finding. This kind of guided by some of the cells that reside there commonly. What we think actually are these subpopulations of fibroblasts. The other thing I'll say is that we're moving forward on not only linking the oral cavity to the rest of the body in adults, which was the last project, but we're starting to understand how it is that the entire, what we call inhalation interface is linked together. The inhalation interface for us is the linkage of the nose, oral cavity, upper airway, including the tonsils and the trachea, on down to the distal tip of the lungs. For us, the really key feature of this is that, and this is a large project that is funded by the Chan Zuckerberg Initiative. Arjun Guha, who's at NSTEM, has been a fantastic collaborator with us to kind of lead this work with a collaboration with India, the United States, and many other countries. But to really understand in children, as the airway, this inhalation interface is experiencing challenge by virus, how it is that they link together. This is the first draft as we've started to get clusters out of many of these sites, including both oral, nasal, even including biofluid cell states, and adding this all to the lung to understand shared and as well as distinct or unique pathways and cell types that reside in these niches. And so using kind of an AI algorithm to predict cell types, including all of these sites, we can find both shared and unique cell types across these spaces. I won't get into this today, there's not really time, but we're finding some really fascinating things that I'll just highlight here, is that if we use spit, which has many, many cell types, including immune cells that I think is missed, as well as the same phenomenon with a nasal brush. These are non-invasive ways to pull cells out from the body that could eventually be used for precision approaches. And what we find is that there's really a diversity in the mouth that's quite different than the nasal sites, that again, as we think about innate and adaptive immune cells, could be quite useful in thinking about kind of overall systemic monitoring in a site that's quite accessible. As you look here in this map here, this orange cluster in the middle here suggests that endotracheal tube, which is something we wash off of a tube that's used for general anesthesia, has a lot of unique enriched populations that I think still need to be explored. That look a lot more like tonsil here in yellow. And so this is an exciting project that has a lot of traction, but requires a lot more analysis, including validation. And so as we think about this idea of kind of an integrated approach, what I'll hopefully get you to the end of the talk is that we hope to move towards the idea of kind of AI-based, machine learning, deep learning-based inferences to the data, and to take that from the overall healthy reference atlas as we started, and to move towards targeting, I think, really available diseases that will benefit from this toolkit. And so what you've seen so far is that we've moved towards some single-cell tissue omics approaches, primarily focused on RNA, although there are other ones that are starting to emerge across a variety of modalities. What I'll really highlight today is the linkage of some of this data to the spatial phenotypes, and then moving towards even thinking about how the microbiome, both viral, fungal, focused on bacteria, can really influence the outcomes on some of these niches, especially at the barrier. And of course, the inhalation interface is nothing but a barrier that's trying to react and I think prevent further diseases within the body. And so as we think about this in terms of an overall focus point, there are bacterial-driven diseases, there are diseases associated with bacteria that are in the head and neck space, as well as lung cancers, glands that include both oral as well as tracheal glands that break down. This oftentimes in autoimmunity, the trachea can break down in asthma, and then even looking at the lungs and the integration of phenotypes like Sjogren's, which is autoimmune-driven, but also things like COVID or cystic fibrosis, which can be quite niche-specific. Leveraging this, we've also started to move towards profiling and I think linking the idea of some of the cell profiling, which is really kind of a border or a fence around some of our cells that might be leveraged as the cost of moving single cell to the clinic, I think, becomes more affordable. The idea of using this non-invasive approach could really help us with some of these other phenotypes like inflammatory bowel disease that I referenced before, but even thinking about things like nasal cancers, or sinusitis or pediatric development overall. And so the kind of comprehensive view here is that these data sets can be built together, they can be integrated, And that will allow us to make inferences as the numbers of this, what we think is really a foundation model of human disease, grows and can be tapped into over time. And so if we're successful about the idea of a multi-omics approach, you have both literature, omics databases, clinical trials, data sets from partners. I would even add here from this reference here from Medium, the electronic health record, linking all these data layers and inference to the genomics, proteomics, and metabolomics. And eventually this leads to an inference on drugs. And we started to do this using some of the data that I'll show towards the end. That eventually, kind of at a human level approach, can take the idea of the human genomic profile and start to predict what might be the best approach. So oftentimes in precision medicine, it is the right patient, right drug, right time. And we think that with this integrated multi-omic approach, so eventually we can start to predict the right patient more often to match the right drug to. So as an example of some of one of the things we're talking about, to do this, we think that we really need biorepositories at scale. Some of the collaborations, again, with Arjun here in India, have been fantastic at starting to collaborate, not just with NSTEM, but with New Delhi, and to bring an India-focused biorepository towards some of our projects. And we have these sites building kind of across the world, because to make these inferences with AI, we need to make sure that we kind of digitize the human being at a scalable feature, so we can extract that information and make sure that we consider the influence of genomics, age, sex on the outcomes of disease. And so in many ways, we've had to innovate to have minimally invasive autopsies. Many patients don't oftentimes want their family members to be disfigured. And so we have a minimally invasive approach in which we can collect many niches from the oral cavity and the upper airway in an autopsy. And for us, as we move forward with this, with the collaboration here with Sao Paulo, we're able to really start to find these micro niches and start to profile them from patients that have both oral, local, as well as systemic diseases. We're moving forward with a project that is able to do this now minimally across the airway down to the distal tip of the lungs. That's getting us at least 20 niches, oftentimes many more than that, to start to understand in a single person how these niches are different at the same time. And so what you might approach from something like that is you would have these spatially resolved micro niches in which you could start to understand how in a same person with their genomic profile, a bacterial event, or let's say an autoimmunity or a cancer can be quite localized and sensitized to that micro niche, which we think influences outcomes. And so to do this, one of the things that we need to start thinking about kind of as a global community is the way we take and leverage our biorepositories and create grids, tissue microarrays for these spatial analyses that I'll show. And we do this now already with glands, the oral cavity being a fantastic place because there are oftentimes small tissue sections that are great for patterning. And so with some of these highly multiplex protein assays, this is one assay that we have in the lab from Akoya called the phenocycler fusion. We can run multiple slides at a time and really high dimensional protein analysis. And what you get out of this is you take the pathology that we traditionally have and use for diagnostics. We move forward towards running these highly multiplex assays, which allow us to understand disease states or neighborhood analysis as everything is grouped together. And then also using something like a MERScope or other technologies that do highly multiplex RNA, in this case a 300-plex, we can start to understand how a cell in a space might express an RNA or other, let's say, genomic assay to really understand how it is that those patterns influence each other and disease states with a spatial context. And so there are toolkits that are being emerged. We have one that's currently under review that have allowed us to quickly assign cells an identity and to start to pattern and understand some of those cell features and those neighborhood features kind of in a tissue-wide setting, both in health and disease. And so I'll show a little bit of that just in a moment. But one of the things that we've started to do with our toolkit, and it's important to think about this when you're evaluating toolkits, is how it performs to the standard in the field. And so ours is an AI-assisted cell identification and phenotyping platform. This is one of the small regions here of a tissue microarray. And within about 10 minutes, we're able to phenotype the cells and give them an identification that's more accurate as we've human pathologically identified these cell types to confirm how the AI is performing against some of the standard in the field, which would be a Seurat approach. The reason this is so important is because if you identify the wrong cell, it's going to have a huge impact in thinking about the way in which we might treat or precision diagnose, or even think about the idea of how we might stratify a patient for a clinical trial. So the translation of this is quite important, and one of the things that we're finding is that by having a human-in-the-loop approach, where the human is guiding some of the AI-assisted diagnostics, we can find cells that are progenitors that maybe we're missing. Or in the context of this right side here, we're finding a huge influx of these orange cells, which are really progenitors here on the T cell side. Of course, T cell progenitor is very important, so they can influence outcomes, thinking about therapeutics, especially in immuno-oncology. The other thing I'll say is that we're learning a lot that RNA alone is really not the only way to move forward. In fact, there's more errors when you use a single modality, and so we've moving forward on running a xenium assay, which is a highly multiplexed assay, again on RNA, but it doesn't have any protein. And so we, again, can use our AI approach and phenotype the cells. These cells, including both epithelial and immune, are easy to find. But we're finding that we undersample some of the immune compartment, especially T cells and B cells, neutrophils. And so we started to leverage both the RNA as well as the protein together, and then using some slide transfer approaches on the assay itself, link the protein and the RNA to the same cell type on the slide. That allows us to phenotype the cells on the protein first, using classic flow cytometry approaches like CD4 or CD8, which would hit a cytotoxic T cell often, or a helper T cell, and then to start to understand their RNA profiles. And I will say that we occasionally get areas where it's tough to align them, and so that's something that we're working on in the lab, is to how it is we take disparate assays and still make inferences and really realign the cells, so we can extract out an individual cell feature with these spaces, even when there's a lot of inflammation with really condensed, let's say, immune foci in the tissue. And so what this might look like here is including both the RNA, but also now you're looking at the protein, which allows us to get much better cell segmentation. And when we link the two together, we can find that we can align them and get pretty good correlation on cell type proportions, leveraging this kind of dual assay approach. And so we think there really is a multi-ohm future that's really required for this moving to clinic, so we can get consistency when it comes to assigning cells and inferring patterns within tissues. Something else that's really important to consider as we all move forward towards thinking about a spatial approach, even if pathology with H&E is the way we move forward, and inferences with a foundation model is really the next steps for any of your clinical or translational pipelines, is that the segmentation, or the box that we put around a cell, is very important. And so there's a number of cell segmentation tools that we started to use. These are deep learning approaches we've used on the multiplexed IF, but we've also used these on H&E, classic pathology stains. And one of the things that you'll see is that we're getting to the place now where using some of the segmentation algorithms out there require training, but can be done on your laptop. And so the first version of this required a high-performance computer, but we're now able to do this. My trainee was doing this at a conference recently, and you can see that over time that the first baseline approach, we're missing a bunch of the cells. These cells are very large and probably counting 2 cells at a time. This one is counting 3. This is not fixed by 20 trainings. It's not fixed by 50 trainings, but we're starting to get better at finding the individual cells at the base. And so not being careful about this over time, what happens is that when you make your assignment on the cells, again, thinking about the translational potential of some of this work, is that you actually are able to lose some of the inferences that are important because cell types are being misassigned. And so we find that when we perform more and more trainings to each of our niches, we improve the number of cell types, including increasing numbers of helper T cells, increasing B cells, natural killer cells, dendritic cells, and neutrophils. So this is very important for thinking about the kind of work that we want to be doing. Is we're still in this kind of Wild West phase of spatial, if you will. And that's a US reference for thinking about how it is that there's really no rules and regulations at the moment of how it is we do this work. And many in the field are kind of scrambling and sprinting to make sure that we can do this in a way that's reproducible and allows us to translate this in a way in which there's minimal risk to the patient. Another way to think about this is once we, again, take kind of classic algorithms or new algorithms, we start to make certain cell types. One of the things that we find is that by running this with a cell pose, which is kind of the standard in the field for our lab and how it is we assign cell types, we make different inferences on neighborhoods and cell interactions that, again, have an important influence on how it is we think about this analysis moving forward. The other thing I'll say is that not just on the cell assignment side, but also in the context of thinking about sample input and what it means to have sample power and inference for some of these precision medicine approaches, is that even when we take unsupervised analysis and start to find immune cell clusters in autoimmune disease, all of these teal cells here and these 4 cases are immune cells. And you see here that case 2 is quite inflamed, and you can already see destruction of the tissue here, where you have these large open spaces. Where here things are pretty well connected, as well as there's very little of that teal. But when we look at a case, this is the 5th case we pulled up, we decided to look at case in both an outer section, we sectioned deeper, and we sectioned even deeper past that. And one of the things that you'll find is that looking at T cells and dendritic cells, the kind of key drivers of this teal phenotype, is that we're missing them at a really high number on the peripheral sections. And the central section is really where the phenotype is. And so this is really important, thinking about the way in which we're going to make a single diagnosis from a single pathology, in this case, an autoimmune disease of the glands. This is really important, considering how it is that how do we have the right number of samples, sections, 3-dimensional pathology to make the inferences that we need. We started to do this now with deeper sections. This again is in asthma, and one of the things that we're finding is that taking some of these phenotypes are translatable. So our toolkit's not an oral toolkit. It's really across the body, as long as the fluorescence from the protein, the fluorescence from the RNA can be quantified. We're able to do this work, and so this is a collaboration with the Tata lab. We're hoping to submit maybe in the next couple weeks. It's finding that utilizing this toolkit in human patients, we're able to find that there's different cell fate changes in some of the acinar cells, which make them mucus. Asthma obviously is a breathing disorder where it's very tough to breathe. And so we can find that some of those mixed cell phenotypes can be plotted, can be understood, can be inferred, and hopefully might be, again, a sign of severity as part of the diagnostic pipeline. And so we're able to do this not just in one space, but really starting to do this across the inhalation interface. So as I referenced, we're starting to think more also about how it is that we can ensure that we're getting the right pathological diagnosis. And so we're starting to create these tissue microarrays that include the spots, where A1 is a unique sample, A4 unique sample, on down the line, and then do this in a way in which we take multiple sections of these tissue microarrays to think about what it is we might do in 3D. And so what you can see here, as I click, this here is really our first approach in which we've done this with 3-dimensional reconstruction. And you find different microenvironments within the context of these spaces. And so we are really thinking about what is the appropriate number of samples to take at different 3-dimensional, we call them Z planes, to ensure that we're getting an appropriate diagnosis for the kind of AI inferences that we need. And so the last thing I'll say in the time we have together is that as we think about classical pathology, again, this might be coming from the upper airway, we think the opportunities really exist in the idea of not just a foundation model, but patient stratification at the level of the clinic, but also at biopharma. So thinking about recruitment, really that right drug, right patient, right time approach. The other thing I think we'll say is that we believe that not only is this diagnostic in terms of its potential, but also prognostic. And so what you have here actually is already something we've been doing in a cancer project with a group that's linked to AstraZeneca in the United States, using an inhibitor of PD-L1. And what we're finding is that there's novel phenotypes at not just the cell level, but the organizational level of cell groups that become a new biomarker for us to think. And we think this could be kind of leveraging the opportunity for companion diagnostics in this condition. The other thing I'll say is that all this RNA that's being profiled at much higher dimensionality than I think we originally thought would happen, at least by this time in 2024. Many of the toolkits are already past 400, 6,000. There's a group that's doing full transcriptome, even though it's quite expensive. This allows us to run certain algorithms that can predict the kind of drug responses. And what you have here in the bottom right is a case of that autoimmunity that is already finding that kind of approved US FDA drugs can be leveraged as a kind of a preclinical screening for the kind of drugs that we might think about using in a patient that's a non-responder to traditional therapeutics. And the last thing I'll say is that it's important to think about how it is you might leverage these kind of drug-to-cell toolkits. But as I hinted at the beginning, we know there's an influence in microbiome. And so one of the things we started to do is look at certain inflammatory conditions in which we can actually metagenomically analyze and annotate at a single cell level the cells, these epithelial cells that have bacteria on them. And one of the things that you can see here is in the health state, having bacteria associated already starts to sensitize these cells to different patterns. They're similar, but they're more aggravated and they're more immune signaling. And we find that dramatically changes depending on the kind of approved drug here that's predicted to work based on the overall annotation of the cell having bacteria or not. And so it's important for us to start to think about not just the single cell phenotypes, the spatial phenotypes, but of course the inter-kingdom or the multi-kingdom phenotypes. It's an exciting world. The amount of information is a lot to handle. And of course, we're going to need other tools like machine learning to really help us make the inferences. They're going to help us get to precision medicine and therapeutics. So our goals moving forward are really take this atlas to start to link these new tissue and cell models as we referenced from the discoveries in the human to kind of leverage a graph-based approach or artificial intelligence to start to interrogate the data in more interesting, hopefully deeper ways. And right now, as part of this kind of biorepository network, what I've listed here in the bottom right, you don't need to read all of these right now, is the conditions that we think are really important for the airway that we can make an approach that would be benefited, hopefully leading to translational potential and treatments for patients that sometimes don't have them otherwise. And so I am very appreciative of your time and the invitation. It's my honor to be here and look forward to answering your questions.